Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Neuroscience Drug Development: How Novel Applications of AI Could Meaningfully Disrupt Traditional Development Strategies. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who register on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario is a leading provider of endpoint data solutions to the clinical trials industry, generating high-quality clinical evidence for life sciences companies. They offer comprehensive evidence generation solutions that combine medical imaging, eCOA, precision motion, cardiac, and respiratory endpoints. For more than 50 years, Clario has delivered deep scientific expertise and broad endpoint technologies to help transform lives around the world. Their endpoint data solutions have been deployed over 30,000 times to support clinical trials in more than 100 countries. Their global team of science, technology, and operational experts have supported over 70% of all FDA drug approvals since 2012. Now it's my pleasure to introduce our speakers, but before I do that, I'd like to welcome and introduce my co-moderator for today's event, and he is Dr. Todd Rudeau, and there he is. Dr. Todd Rudeau brings strong organizational leadership as Clario's Executive Vice President and Chief Medical Officer with over 2 decades of clinical cardiology and pharmaceutical research experience focusing on drug safety. His work centers on developing scientific and regulatory strategy while advancing innovative technologies that strengthen clinical trial rigor and improve the customer, patient, and site experience. Next, I'd like to welcome and introduce you to Dr. Mark Oppler. He is the Chief Research Officer of eCOA Neuroscience at Clario. Hello there, Mark. And Executive Director of the PANSS Institute, leading research in psychiatry and clinical measurement. He has served on the faculties of NYU and Columbia University and is a widely published researcher and developer of multiple clinical assessment tools. Next, I'd like to welcome Alex Boudreau. He is the Director of AI at Clario. Hello there, Alex, where he leads the generative AI organization and oversees deployment of the company's multimodal Gen I platform across cloud, AI engineering, and fundamental research. He brings deep experience from leading high-impact initiatives in deep learning, speech analysis, fraud detection, conversational AI, and medical imaging, rather. Next, I'd like to welcome Dr. Joel Shearer. And there he is. He is Senior Director of Imaging AI at Clario, where he leads initiatives at the intersection of medical imaging, artificial intelligence, and clinical research. With over a decade of experience and a PhD in medical image processing, he specializes in developing and translating quantitative imaging and AI solutions into robust, regulated tools for global clinical trials. Now, with the introductions out of the way, it is now my pleasure to pass the mic over to Todd. So, Todd, when you're ready, you may begin.
Great. Well, thank you, Sonia, for all the very kind introductions there. Thank you to all of you, our quite sizable audience that's joined us today as well. Great to be back for another monthly educational webinar today. Really excited about this one in particular, as there's well, there's quite a bit we've packed into this session, but focusing on novel and really clinically meaningful applications of AI, specifically though in neuroscience development programs. You can go ahead to the agenda. We'll take you through this today, starting off first with a general overview of our responsible use principles. So the framework in which the AI-enabled tools are all developed. I'll cite a few examples of these. And then afterwards, Dr. Mark Oppler is going to speak to digital biomarker development, some really innovative applications, specifically in psychiatric drug development. Alex will follow, continuing on the theme of psychiatric drug development, but pivoting to a description of how AI can enhance the assessment of interview quality. The case study he conveys will focus on major depression, where a clinician-reported outcome is the primary endpoint, and happens to also be quite subject to variability, so we'll hear more about that. And then lastly, we're going to see some particularly interesting applications of AI to neuroscience imaging. So that's often secondary, but occasionally primary endpoint in neuroscience studies as well. At the end, I'll wrap us up and lead into a Q&A session. Please do send in your questions as we go here using the chat function. We'll be sure to cover them at the end. So I can go ahead to the next slide. Just to get an understanding of this audience's perspective before we started, we asked you all by survey during the registration process and on LinkedIn as well, where could AI have the greatest impact on improving neuroscience trial outcomes? So here's the results of that survey, and thank you all for your responses, of course. The 4 choices we gave, first was improving data quality and consistency across sites, so sort of a general data improvement concept. The second was novel biomarkers from voice and speech, so the digital biomarkers piece. Third was reducing rater variability and improving assessment quality. That's referring back to that ClinRO that I mentioned before. And then last was unlocking deeper insights from neuroimaging data. So kind of beyond what a human can do. Pretty even split, as you see here, whether that means we designed the survey well or perhaps means there's just so much potential here in different areas. I suspect, by the way, even though the voice and speech biomarkers got a slightly lower percentage by the end of this, many of you may have changed your tune because there's some really innovative stuff happening there. So go ahead to the next slide. I think it's important we start here as this is the foundation or framework in which all of the AI-enabled enhancements for neuroscience development that we'll be talking about today were developed. And this is a slide that hasn't changed since 2023, and that is a very good thing. That's when these principles were originally defined in this structure, although in reality, many of them predated that just without the defining framework as you see it here now. But to keep us on track time-wise, I'm not going to go into a ton of detail here, but did briefly want to at least touch on the 5 principles that are outlined here. So starting at the top left, minimizing AI-induced bias. This is all about cognizance of selecting a training data set and a process for training that considers the potential use cases holistically. So how will this be used in the future? If this is possibly going to be used in pediatric studies one day, we can't just train it on adult-only data, for example, right? Making sure that the algorithm has everything it needs to perform as desired. Ensuring accountability in the top right. So this is all about maintaining transparency to how a tool was developed and how it's being deployed. So ensuring that the end user of that algorithm or that tool knows the limitations or potential limitations and can adjust their use of that, accordingly. Considerations around IP and data privacy rights is next. So compliance with global data privacy laws, using aggregated and anonymized data in all the R&D work that we do, ensuring no traceability back to any individual subject, any individual sponsor or study. That's what this principle is all about there. At the 6 o'clock position next is the emphasis on deploying appropriate risk mitigation strategies. So again, the focus here is, how can we ensure that the AI-supported results are trustworthy? And largely this involves understanding level of risk and then including human oversight wherever most appropriate, particularly though with any tools that are going to be touching human data during study conduct. Uh, Last but not least, cognizance of the regulatory landscape continually evolving. This is just ensuring that we maintain appropriate compliance with global regulatory policy and awareness as policies evolve. On the next slide, there's some examples of tools we've developed and deployed into studies that we're currently supporting. By no means a comprehensive list, and certainly not the purpose of today's webinar, but I did include just a few examples here, and they fit into three broad categories. Tools to enhance data privacy for participants, tools to optimize workflows, and then tools that are built to maximize data quality. And it's this last pillar where I really believe we can have the most meaningful impact in clinical trials, improving the precision and accuracy of data that ultimately is going to be feeding into those final analyses for the clinical study report. In the first pillar, the privacy piece, these are tools that simplify the site experience. So uploading a document that inadvertently contains PII, the tool can automatically recognize and redact this without any steps whatsoever for the site staff to take action on. It is subsequently reviewed and confirmed later by central staff, but not any burden for the sites. In the process optimization pillar, these are both internal and external facing solutions. Streamlining study startup, for example, by accelerating charter document development, applicable for imaging and ECOA solutions as well. And in that last pillar, as I mentioned, applications to improve data quality, this is where it gets super interesting and impactful. AI supporting a radiologist, for example, measuring lesions, promoting consistency from one time point to the next, and across different readers and different subjects all in the same study. You can imagine, improving the precision of measurements, reducing noise in the data. All of this leads to narrower confidence intervals during the statistical analysis phase of the study, and therefore a higher likelihood of differentiating signal from noise to confirm, a statistically and clinically meaningful result. AI can help us and does help us in quickly reviewing ECG waveforms. So in a 24-hour heart monitor or Holter recording, identifying low quality areas of the recording that may not be suitable for the measurements and the analysis that's intended. And if so, doing so in a timely way so that it can be corrected, right? So not just looking at data quality days or weeks or months after it's been acquired, but doing so in a time-sensitive way that can actually help improve any gaps. So to take it from here, I'm going to hand over to Dr. Mark Oppler now, who's going to take the next maybe 15 minutes or so to speak about what, as I mentioned, is an incredibly innovative and interesting application of AI in neuroscience, and that's the role for applying AI to digital biomarkers, and specifically in psychiatric drug development. So, Mark.
Thank you so much, Todd. Appreciate the kind introduction. So let me start today first by reflecting on something that Todd was kind enough to outline, which is that we have several problems of reliability in psychiatry and in neuroscience broadly. But in psychiatry specifically, one of our biggest challenges around reliability and confidence intervals has to do with the measures that we use, particularly our endpoints and our measures of efficacy, sometimes as well as safety. So just to outline for those of you who aren't steeped in this literature, in psychiatry, our measures tend to be somewhat old and creaky. They are typically developed in prior eras, many of them decades old. They're not necessarily quick or easy to use. They're not tools often developed for high throughput research on an industrial scale. Many of them are quite subjective, and they're based on the clinical interview process, which is again a venerable and time-tested method of interacting with, evaluating, and collecting data on participants in clinical trials, but it's very time-consuming and it's very prone to error. So for as long as I've been working in this area, there's been a discussion happening where everyone admits to the problem. They say, the scales we have, the instruments we have, the endpoints we have are terrible. Surely we can come up with something better. What about digital tools, wearables, apps, and so forth? As we'll see on the next slide, the answer to that proposal, particularly when you use AI to try to develop these things or to come up with better methods of assessment, the results are a little bit mixed. And across the board, I think there are some bright spots, but generally speaking, there's a lack of consistency in the findings. Our efforts to use AI to try to develop better techniques clinically, uh, as we see here in one of the classic examples from the early efforts with IBM Watson, have produced again very inconsistent results. Sometimes this is due to the fact that when we use AI to try to improve processes, outcomes, or procedures in healthcare or in research, we're often training our models on very limited and sometimes hypothetical data sets. The result is an ungeneralizable outcome. And I think this problem writ large also illustrates one of the issues that we have in the development of digital endpoints, like, for example, digital biomarkers. On the next slide, there's, I think, a nice quote that I like to reference periodically. It's getting older, but it's still true. This is a quote from a 2018 paper in Clinical Pharmacology and Therapeutics. That currently the field is full of enthusiasm, but more data are needed to from rigorously designed studies to displace the hype. So, 20 or plus years of efforts to create digital biomarkers in psychiatry, aided recently by AI, has resulted in a lot of different tools. But for the most part, these have stayed exploratory. These have stayed in the realm of shiny toys. And their impact, to be perfectly frank, has been pretty limited. So, myself and others here at Clario, in collaboration with our academic partners, have been working hard to try to come up with better approaches and to overcome the limitations that we've seen in the field so far. Why don't we go to the next slide? First, I think we've started by trying to outline what's the matter with our novel endpoints and tools. What's the matter with biomarkers, especially digital biomarkers in psychiatry? By focusing on these specific challenges, we're trying to focus our AI development process in a more productive way. The first challenge that I think everybody recognizes is that these tools, whether they're wearables or anything else, are often very challenging to implement. The introduction of every new device, every new process, every new procedure in a clinical trial tends to add burden, both to participants as well as to investigators. The second point about a lot of the digital biomarkers in psychiatry is that some of them are very complex and hard to analyze. Some of this arises out of the use of black box AI techniques, coupled with data sets that don't generalize well. The third issue is often that the purpose behind these tools, why are we using them, what problem are they solving for, is often unclear. I think between these three challenges, we've seen a big barrier to adoption as well as limited impact. Let's go to the next slide. What we're trying to achieve, I think, is best illustrated by a quote from an investigator who we often turn to when we're looking for guidance. We asked this individual and several other people, what do you want to get out of a digital biomarker or a novel digital endpoint? And they said, almost unanimously, we want it to be invisible. In other words, we don't want it to feel burdensome. We want it to be useful to actually solve a problem rather than create one. And we'd like to be meaningful, to actually tell us something we don't already know or do something we couldn't already do. So to that end, our drive has been to search for digital endpoints and biomarkers that are simple to implement, that involve automated objective analysis. In other words, that don't require complex algorithms in order to analyze, understand, and interpret. And third, we're looking for things that actually have a strong relationship to the scientific objectives of our studies. So these have been our guiding principles. As we'll see in the next slides, this has resulted in a tack that I don't think any of us necessarily could have predicted. But it's led us, I think, in a very interesting direction. The example that I'm going to illustrate for you today is the development of a marker called speech latency. So, as I mentioned before, most of our endpoints in psychiatry are based on clinical interviews, the interaction between a clinician and a study participant, driven often by a scripted or a semi-scripted interview. Recordings of these interviews have been studied for a very long time, as well as recordings of free speech, diaries, audio diaries, and other forms of recorded speech from patients and participants in studies in psychiatry. These have been studied in many, many pathologies and disorders and therapeutic areas, including depression. One of the things that we found when we were extracting a range of vocal biomarkers from recordings of clinical interviews was there was one that kept coming up over and over again as being potentially important. And that was something called speech latency. As you see in the graphic here on the screen, this is a recording of a clinical interview of the MADRS, the Montgomery-Åsberg Depression Rating Scale. The blocks in red are the times when the clinician or the rater is speaking, and the parts in blue are the patient. And those little interspersed sections in yellow are the pauses between the clinician finishing asking the question and the patient's time to respond. That measurement of time, averaged across the course of the interview, can be calculated as speech latency or speech turns latency. It includes, amongst other things, a measure of the patient's time to react. Let's go to the next slide. When we looked at speech latency, this form of speech latency, reaction time, in data from a large clinical trial of a mood disorder, we found something pretty astonishing. First thing we found was that by modeling just this very simple calculation, the patient's time to response after the clinician asked them the question, was highly related to the primary outcome in the study, the Montgomery-Åsberg Depression Rating Scale total score, the MADRS. But in addition to that, when we looked at these turn latencies, these reaction times across every country in the trial and every language spoken, and this was, as I mentioned, as I should mention, a large global trial involving 7 different countries. We found that remission in depression, in other words, clinically meaningful reduction in scores on the MADRS, was highly related to speech latency, in that patients who were still depressed or whose depression was prominent and clinically significant had much longer speech latencies, much slower reaction times than patients whose depression had fallen to a clinically meaningful level or who had remitted. Across every country, across every language, patients with less depression and lower scores were consistently quicker in their responses. They sounded and reacted in a way that was more normal. Let's go to the next slide. But the thing that really, I think, woke us up to what we were looking at here was this. When we stratified, when we divided the patients in the study into 2 groups, those whose speech latency, or as we started to call it, their vocal biomarker phenotype. When their vocal biomarker phenotype at screening, prior to randomization into the trial, was quick and normal, even if their MADRS total score was high, we labeled them vocal biomarker negative. They're represented in the graph on the left. Those patients who at screening had longer latencies, indicative of higher levels of depression, we call them vocal biomarker positive. They're seen on your screen on the left and vocal biomarker positive. So vocal biomarker negative on the right, vocal biomarker positive on the left. These graphs demonstrate the changes in the MADRS total score in the clinical trial I was referring to. The black line shows you the placebo group, patients on placebo. The green line show you the patients on 2 different doses of the drug that was under investigation. And what you probably see immediately is that the vocal biomarker positive patients show a robust response to the study drug, but a meaningfully less response to placebo, shown in black, whereas the vocal biomarker negative patients on the right have no difference whatsoever. Um, to sum up here, vocal biomarker positive patients showed about a 2 to 3-fold increase in effect size. In other words, in these patients whose speech latencies were long and whose moderate scores were high, everything was aligned, objective and subjective, we had a very powerful demonstration of clinical enrichment in the study. Whereas on the right, vocal biomarker negative patients showed no difference between drug and placebo, essentially generating a null result. Let's go to the next slide. Just to fast forward, in studying this over the years here at Clario and with our colleagues in academia, we found that similar functions and variations on the speech latency feature, when applied to other disorders like schizophrenia, show similar powerful effects. Vocal biomarker positive patients in schizophrenia studies show robust responses to antipsychotic treatments. Vocal biomarker negative patients show a much more muted response that sometimes takes a very long time to come to any meaningful result. This is not only true in overall efficacy of these drugs, but if we go to the next slide, you'll see that in very difficult to treat phenomenon, such as negative symptoms in schizophrenia, we see very similar results. Again, depending on the phenotype of the patient prior to randomization into the study, we can predict with reasonable certainty the likely outcome. Let's go to the next slide. Beyond the use of this particular feature of voice as an enrichment tool in clinical trials, we are also coming to believe that it may serve as an endpoint and demonstrate sensitivity to change. Going back to the results of the depression study that I talked about before, we also noticed that in patients who remitted in that trial, when the two arms of the drug showed very significant changes actually in their speech latencies across the study. This suggests that some of these markers serve not only as trait-like markers and enrichment tools, but may also be state-like and form novel endpoints to measure drug efficacy. Let's go to the next slide. To sum up here, I think that we have an opportunity to move beyond the use of these tools as exploratory, go from shiny tool to useful by using audio and video that we routinely capture on our RECO platforms, subjecting these tools to near real-time analysis. We can use these things, these novel measures, to enhance signal and for other applications that may actually move our research forward. I'm going to stop there. I think that's all I wanted to say today, and we can go on. I will now pass the microphone to my colleague, Dr. Alex Boudreau. Alex, over to you.
Thank you, Mark, and hello, everyone. It's really a pleasure to be here today. Just to recap a little bit on some of the things that Mark has mentioned, there are really some unique challenges in psychiatric studies. Right? First, they rely on subjective endpoints for assessing the safety and efficacy of treatments, which involve patient-reported outcomes and clinician-reported outcomes, which capture information and data on the symptoms of the trial participants rather than direct objective biomarkers. Right? So in the end, this introduces bias and variability in the study, which we want to avoid. If we move on to the next slide. If we look at one type of clinician-reported outcome, COA interviews, these are semi-structured conversations held between a clinician or rater and trial participants, and as mentioned, used to collect data and information on how the patient feels and functions throughout the study. And there are multiple standardized instruments that can be used in studies, including MADRS, as mentioned previously, which stands for Montgomery-Åsberg Depression Rating Scale. And as the name implies, this is an assessment used for measuring the severity of depressive symptoms in individuals in the studies. If we move on to the next slide. Now with MADRS, we have what we call Sigma, which is the structured interview guide for MADRS. And what this provides is a well-defined and standardized assessment with predefined questions, branching logic, follow-up questions, with the objective of the rater to assess the severity of the symptoms across 10 scale items from reported sadness to suicidal thoughts. If we look at one example for scale item number 2 on apparent sadness, right? Here the rater, as he has a conversation, or as they have a conversation with a participant, they would rate the symptom from 0, having no sadness, up to 6, looking miserable all the time. If you move on to the next slide, as you can imagine, right, these are complex conversations. They're lengthy and include a lot of information. And in fact, they take, on average, 21 minutes to conduct at baseline. And in fact, they also can extend well beyond 21 minutes to 40 or even beyond throughout the study at different time points. Now, this introduces a series of challenges around data quality. So you have the rater that it can introduce variability and subjectivity in the assessment, whether it's intra-rater or inter-rater variability. For instance, they may deviate from the established standards for the assessment. Right? For example, they might not follow all of the questions or not ask the questions in the intended form from MADRS to other assessments. Or they may go and do an incomplete assessment, meaning they may not go through the entire assessment, not ask all the required questions as part of the instrument. Now, if we look beyond administrative challenges, there's also standard operational challenges, such as low quality recordings, incomplete recordings, and other quality issues with audio. Now, since COA interviews are actually really crucial instruments, right? They're the primary endpoints for determining the efficacy and safety of treatments in those psychiatric studies. There's a process called independent review where you have highly calibrated experts that come in and manually review the recordings of those interviews to ensure that they're of high quality, that they're completed per the assessment, and they're completed in full. You might imagine that this is a high effort process, and in fact it is, right? If you think about a multinational setting where you have to study across sites in different countries at a global scale, you have recordings in multiple languages at different sites, and you have these experts that need to come in and review manually those interviews. So this is highly time sensitive, costly, and for that reason, typically, only a small percentage of all those recordings are reviewed by these calibrated experts, say at 20%. If we move on to the next slide. To compound the challenges with these types of studies, the independent review process itself is also potentially subjective, right? And to mitigate those challenges, there are various standardized approaches to enhance the consistency of that expert review to improve, in the end, drug placebo signal detection, right? So to improve the quality of the data and of the review. So you might ask yourself, and you might think this is probably prime for AI innovation. You're absolutely right. We can leverage AI here as a really powerful enabler for enhancing study outcomes. So if we go to the next slide. There are different approaches and different aspects of these interviews that we can enhance with AI. And one first area is around the depression scoring. So if you think about MADRS as the instrument, you can train a model to predict the severity of symptoms for all scale items from 0 to 6 or as a whole, right? So you have an AI model, for example, that can say, for this interview, I score it as 42, which stands for severe depression. Based on the instrument. So on one hand, you can do that. On the other hand, you can also look at overall interview quality. So having AI score the quality of the interview to ensure that that it follows the instrument, that it's completed in full and is of high quality. Now, this second aspect is actually more challenging than automatically detecting the score of depression, right? Because here you have to teach AI to understand the nuanced factors of what is a quality interview. So many challenges to have a holistic solution there. If we move on to the next slide. So if you look back at implementing an AI solution that performs that effectively and reliably, what's important to note here is that this must be addressed holistically, right? So your system needs to address the interview analysis holistically to provide meaningful signal. And only providing a depression score is not sufficient, because you need to have indication whether the interview was properly administered and whether your patient or the participant has the right phenotype. So if you remember, if you recall, as Mark Oplo mentioned, you can have a VBM positive or VBM negative participant, and having true depressive symptoms and is actually really critical here. Otherwise, automatically detecting depression score may provide more noise than actual meaningful signal. Now, to support that, what's really critical here is to have a really large and well-defined expert data set, right? Expert defined data sets as a ground truth to make sure that you can actually properly train your system and that you can properly validate it. If we move on to the next slide. So what we built here at Clario are actually two systems. We've built a system for automatically extracting the digital biomarkers, in this case speech latency as a vocal biomarker, which allows us to identify the phenotype of the participant as well as other metrics. And we've also worked on building an automated independent review system that provides the depression score automatically, but also analyzes the full interview from a quality and completeness perspective as a calibrated expert would. So if we move on to the next slide, if we look at how this works at a high level, right? So you have a site where the interview is conducted. That interview is recorded. This audio recording is then sent to our cloud platform for analysis, where it is actually processed in near real time. And at the output of that analysis, we have a report, also some flags that indicate whether the interview is of high quality and that it can be reliably used in the study, or otherwise, if it should be reviewed through human experts. If we look at the technical aspect of how this works, so on the first step, once we receive the audio file, there is multilingual speech analysis that is performed. That includes speaker diarization, which is the process of splitting the different speakers in an audio session, right? So you can understand if there are one or multiple speakers, and you can separate the audio alongside the acoustics of the different speakers. Now, the next part is to do multilingual transcription, which means essentially converting the speech to text so that it can be further analyzed downstream. Now, all of this information is sent to our in-house custom-built agentic AI engine, which takes the full interview transcript, so that's speech to text aspect, as well as the labels of the different speakers. So you might have speaker 1 and speaker 2, if there are 2 speakers. And the system then automatically detects which speaker is the rater and which speaker is the participant, to be able to understand and analyze the contents of the interview meaningfully with the right roles. Now we have also an AI agent that actually pulls in information about the assessment, so in this case MADRS, as well as the criteria used by human expert, right? So the independent reviewers that perform a calibrated review of those interviews. So the system essentially emulates human experts in reviewing the overall interview to assess the quality and the completeness, as well as performing the overall automatic depression scoring. So all of that information then is processed and generates a report at the end. and provides a flag if a interview should be reviewed manually, if it's a higher risk or lower quality data, for example. So what this provides is the opportunity to process 100% of the interviews in an automated fashion using near real-time AI technology. We move on to the next slide. Just to conclude here, to highlight some of the key takeaways from this session. The critical ingredients here for providing a highly reliable and accurate AI system for automated interview analysis requires, first of all, to have a really high quality and expert-created ground truth data set. This is really critical. You cannot use mock data and mock interviews. You need live interviews. You need experts that understand the process and the instrument and that provide the right labels at length. You also need a system that is able to do the depression scoring automatically, as well as the independent review automatically to assess holistically the quality and completeness of the interview. And then finally, you of course need extensive scientific validations to ensure that the system is reliable, accurate, and ready for production. So with that being said, I will hand over to my colleague Joel.
Thank you, Alex. I will now tell you how we apply AI to the imaging part of neuroscience trials. So at Clario, we apply AI throughout the entire life cycle of imaging in clinical trials, going from image triage all the way to quantitative image assessment. In order to do this reliably, we deploy AI models centrally to a validated platform that complies with all the relevant AI, privacy, and computer security regulations. So as I was saying, we start with image triage, where AI models help us automatically label images as they arrive from the site, sorry. And this helps us identify submissions that may be incomplete, flag them, and report them in a timely fashion in order to maximize the chances that complete data sets will be acquired. Next is anonymization, where AI helps us remove the faces from high-resolution CT or MR images, which could otherwise be used to re-identify patients. Next is QC, where AI assists in identifying submissions with problems, such as submissions from different patients, or acquisitions with quality concerns, such as incomplete coverage or artifacts. Again, the goal is to help flag these cases on time and not replace human oversight, but instead identify submissions with problems, flag them, and assess them with priority. And finally, the most important aspect of AI in clinical trials is using it to help with image interpretation. And this can range from reader assistance to more involved use of AI, all the way to new biomarkers that are enabled by AI. And so for this presentation, I'll focus on this last AI-assisted image interpretation part. So on the next slide, we'll start with read assistance. Usually, read assistance involves image segmentation, meaning the automated delineation of organs or lesions. Traditionally, this was done using traditional image processing technologists or directly from the reader. This is, of course, a very labor-intensive process. It can lead to fatigue and increased variability in the assessment. So AI can be used to automate the bulk work of this delineation and only leave minor edits to the technologist or the reader. And this allows the reader to focus on what is really important, the interpretation of the image. However, if we want these read assistance models to be successful, they need to be deployed thoughtfully. And this includes three key success criteria. The first is pre-deployment. Before we even think about deploying AI in a clinical trial, We need thorough validation, meaning clinical validation with medical experts, and large-scale statistical analysis. Regular software testing is not appropriate for AI models. We need large-scale analysis since the models are not deterministic in order to assess at scale their performance, their behavior on different populations. And lastly, but importantly, we try to publish our results in scientific conferences or journals in order to increase our trust in the models. Next is the integration pre-trial. Here we focus on the added value for the trial instead of deploying AI everywhere without thought, and we train and work with readers in order to ensure the adoption of the model. And finally, during the trial, continuous monitoring is used to quickly identify issues as they may arise. For example, data drift could happen, and continuous monitoring can help identify it earlier and react accordingly. Next slide, please. So a first example of read assistance that I'd like to show you is an example in metachromatic leukodystrophy, which is a rare disease characterized by progressive white matter demyelination. So as you can imagine in this disease, the quantification of demyelinated white matter on MRI is a key biomarker, but it is particularly difficult and labor-intensive to assess. So in order to help with this problem, we trained an AI model from a set of semi-automated segmentations. The trained model shows great agreement with the previous segmentation with a dice index of over 90%. It also shows good association with clinical markers, as well as consistent longitudinal behavior. But more interestingly, the model shows greater association with the clinical scales compared to the semi-automated segmentations that were used in this training set. And this shows that AI can generalize beyond what is shown in the training set and regularize the results. Next slide, please. So here I'll show you two more examples of read assist models. The first example is for stroke, and specifically midline shift, which is a very important biomarker in stroke, showing how the midline of the brain shifts in response to intracranial pressure. And interestingly, this marker is not segmentation based, it's based on the detection of key salient points, but AI can also be used here. And here for this model, we trained a model and it showed strong agreement with a radiologist with a kappa over 0.8. On the right, we have an application to cerebral adrenoleukodystrophy, or CALD, which is rapidly progressive inflammatory destruction of cerebral white matter and seen as hyperintense lesions on FLAIR. Here as well, the model shows great agreement with the readers with a DICE index of over 93%, and a good agreement with the semi-quantitative rating scale that is used traditionally. The model enables full volumetric assessment of the lesions and shows greater agreement with a blood marker of neuroaxonal disease. So it shows greater agreement with this blood marker than the traditional semi-quantitative scale. Next slide. So as we've seen, AI can help with estimating biomarkers for efficacy, but it can also help with patient safety. Amyloid-related imaging abnormalities, or ARIA, are a well-known adverse effects of amyloid beta-directed treatments in Alzheimer's disease. They can have two forms, ARIA-E or edema, and ARIA-H for hemorrhage. These can be difficult to identify on MRI. They can be very obvious or they can be subtle. And while most of ARIA events are asymptomatic, they can be serious and even fatal in cases. So it's very important to identify them as soon as they occur in order to process the patient appropriately. So for this purpose, we trained two models, one for ARIA-E and one for ARIA-H, and these models are longitudinal. They take two inputs, a baseline scan and a current scan, and only identify the new lesions. As you can see in the table on the bottom right, our AI results show great sensitivity, but also importantly, good specificity, which is important in order not to burden the readers with irrelevant information. Finally, for this model, it was important to integrate the model in a way that does not bias the readers in order to maximize patient safety. So the way we integrated the model is to have the reader work in parallel with the AI. The reader is not shown the AI results when they do their work. However, when they're ready to submit the work, the system will automatically flag cases where there is significant discrepancy between the AI results and the human results, and ask the reader for confirmation in these cases. Slide. AI can also go beyond read assistance and act as a catalyst for removing study constraints. A first example of this is improving statistical power. We trained a brain segmentation AI model, and we saw a 25% reduction in required sample sizes for detecting atrophy over traditional image processing methods. And this goes beyond image segmentation and volumetry, since brain segmentation is used everywhere in imaging for other biomarkers such as DTI or fMRI. Next, AI can speed up turnaround time, especially in high-stakes scenarios such as eligibility, where time is of the essence. By reducing human review burden, AI can help improve our turnaround time and help us give meaningful results quickly enough to take operational decisions. And finally, AI helps us support heterogeneous populations. Here on the right, we have an example of a pediatric case. Pediatric brains are notoriously difficult to segment because they evolve quickly, the anatomy changes, the contrast changes, and data is hard to come by. But by leveraging AI over synthetic data, we can train models that generalize well, even for these difficult populations, and get great segmentation and really meaningful improvement, as you can see, over traditional methods. Next slide. Finally, AI can help explore novel quantitative biomarkers and help us go beyond what is possible for humans to do. And here I would like to show you the example of enlarged perivascular spaces, which are a marker of small vessel pathology in neurological diseases and provide complementary information over more common imaging endpoints. These perivascular spaces are challenging to quantify because patients can have up to hundreds of enlarged PVSs, which is impossible or nearly impossible to quantify by hand. So instead, readers would typically do a semi-quantitative assessment by looking at one slice, but this only offers part of the picture. By leveraging AI, we can do a full volumetric assessment and get a complete picture of EPVSs in patient brains. Next slide. So in order to do this, we trained an AI model to segment EPVSs over the whole brain. This model was trained to use only 3D-T1, so this removes the dependency on 3D-T2s that are usually used in addition to the 3D-T1s to assess PVS. The model shows great correlation with the semi-quantitative human results, as you can see on the bottom, but it offers a more granular count of PVSs by giving a full count of all enlarged PVSs over the patient brain. The results also show great correlation with clinical parameters. Next slide. So to summarize, for imaging, AI enables higher quality and reproducibility in neurology trials, as well as streamlined operations. Successful AI implementation requires thorough validation, collaboration with readers, and careful monitoring throughout the trial. AI can lift operational constraints, for example, by reducing required sample sizes, and AI can unlock new biomarkers that were previously hard to quantify. And with that, I'll hand it over to Dr. Rudo.
Yeah, excellent. Well, thanks, Joel, and really interesting applications there, certainly, and appreciate you taking us through them. One slide here just to quickly close us out and transition into Q&A. The takeaways are listed here. Just to quickly summarize, we've heard, I think, a very compelling set of examples for the impact that novel applications of AI can have in neuroscience drug development. So digital biomarkers, including vocal biomarkers and speech latency in particular, adding meaningful insights now within studies. Examples were shared both with schizophrenia and depression studies today. In addition, using AI not just to extract signals of disease status or disease drug efficacy, but also fundamentally using AI to assess the quality of that data. So, identifying underperforming sites or individual raters, addressing those areas of underperformance mid-study rather than learning of deficiencies at the end. I think you can imagine how that really can move the needle. And then, as I just mentioned, from an imaging perspective, super innovative applications there to different modalities and how these can support the neuroradiologists and provide insights that otherwise wouldn't be feasible by conventional methods. So let's jump into some Q&A here in the last few minutes. Thanks to those who have taken a moment to submit questions. I'm going to invite all of the speakers, Mark, Alex, Joel, why don't you come back on camera here if you would. This first one, Mark, I'm going to point your direction if that's okay. I actually think it's a super interesting question. Should we be using vocal biomarkers as inclusion criteria in psychiatric drug development protocols?
Just throw me in right in the deep end, Todd.
Yeah.
Thank you. No, that's a great question. I think once upon a time I would have said, maybe in the far distant future when we have flying cars and robot butlers. I don't think it's that far off, and I think it's finally probably at a point where it's worth considering. There is, of course, an FDA set of guidelines around enrichment in clinical trials that I think might be a great paradigm for that. And I would say, the time may finally have arrived. Anybody who's actually thinking about doing that, however, needs to take into account the regulatory considerations as well as the scientific and clinical ones. People are welcome to see me after class if they're genuinely interested in discussing that approach. I think it's a very interesting question.
Yeah, I agree completely. Alex, I'm going to send this one your direction, and I think, interestingly, you could take it as general or get actually pretty specific into the cases or the case you described. But so this question is about challenges to developing a reliable system for automated independent review. So, as I would interpret this, it's basically, you set out with a task, and how do you know this is reliable? How did you come up with the training process for what you talked about today? So, good question there.
Yeah, it's a very good question. I can speak at length to that, but I'll try to keep it short. So what's really interesting about the automated independent review problem is that you need to, interview analysis is really multifaceted, right? It's a multifaceted problem that includes details and information from different angles. So it's not just about audio quality. It's not just about if the interview was conducted in full. So you need to build a system that really understands what the instrument or what the assessment is about, right? What it consists of. Also, whether it was properly administered, making sure that the rater is not leading in any way and providing objective data as much as possible. It's also a system that needs to understand the flexibility, where there's permissible flexibility. When you think about humans, how they review, they make judgments, and they make calls to whether or not that's okay for this study. So but you need a system that understands those nuances, which is quite complex for AI, right? So it's overall more involved than your traditional machine learning, where you train a model to predict a label or a class, right? So this is really holistic analysis and understanding of those multimodal interviews. And for that, you need some really strong data sets, ground truth data sets defined by experts calibrated experts that understand how to label this information from their perspective, from their domain expertise. So we need to work closely with our scientific partners in that process. So happy to speak some more about it at some other time.
Great, thanks. Yeah, spot on. I think this concept of permissible flexibility is interesting and maybe frightening at the same time, that we're actually able to have a machine not think in a black and white way, but to make a judgment call essentially. So, all right. Thanks again, Mark, Alex, and Joelle for your contributions. Sonia, I'm going to turn back over to you. I realize we are at the top of the hour, and I'll have you close this out.
Okay, well, thank you very much for answering those questions. And just like Todd said, we have reached the end of the Q&A portion of the webinar. Thank you firstly for such a great, inspiring, and a great presentation there. I'm sure everyone enjoyed it. For those of you who are interested in hearing more from Clario, you can register for their next webinar, Risk-Based Monitoring in Clinical Trials, Improve Oversight Through Intelligent Review of Digital Endpoint Data, scheduled for Thursday, June the 11th. 2026, using the link I've sent in the chat box and also this QR code right here. Okay, thank you so much for that. We'll go to the next slide here. And if we couldn't attend to your questions, rest assured the team at Clario will follow up with you after this presentation. And if you have any further questions, direct them to the email address you see there on your screen, science@clario.com. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve our webinars. Additionally, I have shared a link to view the recording of today's event in the chat box, which you can share with your colleagues once they register for the recording here as well. And for those of you watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to such features as viewing the recording and more. Now, please join us in thanking our speakers for their time here today. So thank you, everyone, and thank you, Todd, for being my co-moderator. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team at X-Talks, we thank you for joining us. Until next time, I'm Sonia Hunt. Please take care and bye for now. Bye, everyone. Thank you. Bye.